Smart molecules, the new logic gates of life. We've already seen how gene editing tools can rewrite our DNA, and how delivery vectors carry those tools into our cells. In Part 4, we explored how external signals like drugs or electromagnetic pulses might one day have limited control over our gene editors and nano implants. But that approach can't scale. Flooding the body with chemicals or energy fields isn't a safe long-term strategy. To really manage thousands of gene packages at once, we'll need something that works from the inside out. That's where smart molecules come in. Tiny logic units that react to local chemistry, change function, and do it all right inside our tissues. Our bodies already use these kinds of molecules to stay in balance. The challenge now is to augment them. Can we create a toolbox of smart molecules, the logical processors that will give us super longevity? Nature already builds logic molecules. Nature has been developing smart molecules for billions of years. They're embedded in every cell and every system. Take the lactose operon in bacteria, a tiny built-in logic switch that controls when the genes for digesting lactose turn on or off. When no lactose is around, the repressor protein clamps down on the DNA and blocks the gene. The system stays off. But when lactose appears, it binds to the repressor and changes its shape. The repressor protein lets go of the DNA, the gene turns on, and the cell begins producing the enzymes that digest lactose. That's pure molecular logic, an input, a decision, and an output, all written in chemistry. The system senses lactose and changes its function accordingly. Building synthetic logic. What nature discovered, scientists are now recreating and expanding. Using AI design tools, researchers can now build a whole range of molecules that make their own decisions, from smart polymers that respond to pH or heat, to RNA and DNA systems that sense molecules inside cells. Riboswitches are a classic example. They're made of RNA that folds into different shapes depending on what it binds. That binding site is called an aptamer, a region that recognizes a specific molecule. When the right molecule attaches, the RNA changes shape, sometimes blocking, sometimes allowing the message to be read downstream. This can happen at the transcription stage, stopping a gene from making its mRNA, or later at translation, preventing the mRNA from making a protein. In effect, the binding molecule becomes a switch that turns genes on or off. Where we are today. In research labs, scientists are already running simple genetic circuits inside living cells. It's still early work, mostly in bacteria, yeast, and cultured human cells. But every successful circuit brings us closer to real molecular control systems. These experiments show that biology can process information, turning genes on or off in response to signals like oxygen, pH, or inflammation. AI design tools are now testing thousands of molecular variations in simulation before any are built. The goal is to link these tiny switches into small, reliable networks that can coordinate how a cell behaves. It's the first stage of programmable life. But where will we be 5, 10, or 20 years from now? The rise of molecular logic gates. In just the last 15 years, the number of molecular logic systems built and tested in the lab has grown from a few dozen to thousands. Research investment has surged from under $2 billion a year to nearly 10, and it's still accelerating. Each generation of designs is becoming more complex, moving from single riboswitches to hybrid, multi-input circuits that can coordinate a cell's behavior. If the current trends hold, the 2030s could bring networked molecular systems, logic boards made of living chemistry. That's when the real-time biological control could begin to emerge, with smart molecules quietly managing repair, defense, and balance inside our cells. The next grand challenge. We're standing at the edge of a new era. Thousands of molecular logic controllers ready to link into living networks. They could coordinate repair, balance metabolism, and guide regeneration. Everything we need for longevity. But there's a problem we can't ignore. How can we be sure these complex systems will be safe and effective once implanted in the human body? That's the next grand challenge. Not just building the hardware, but programming internal networks of molecular switches that can work together and adapt to changing conditions. And just as important, every design must be personalized, because what works in one person may fail in another. In the next episode, we'll confront that challenge head on, defining what it will take to map this vast design space and bring this technology to reality in years instead of decades.